In order to move the fingers, the nerve impulse must travel a certain path from the brain to the muscles that carry out the movement of the fingers. Excluding reflexes, all skeletal muscles contractions occur as a result of conscious effort originating in the brain. The brain sends electrochemical signals through the somatic nervous system to motor neurons that innervate muscle fibers. The connection between a motor neuron axon terminal and a muscle fiber occurs at a neuromuscular junction site. This is a chemical synapse where a motor neuron transmits a signal to muscle fiber to initiate a muscle contraction. The sequence of events begins when an action potential is initiated in the cell body of a motor neuron, and the action potential is propagated along the neuron's axon to the neuromuscular junction. Once the axon potential reaches the end of the axon terminal, it causes the neuromuscular neurotransmitter acetylcholine from synaptic vessels in the axon terminal. The acetylcholine molecules diffuse across the synaptic cleft and bind to the muscle fiber receptors, thereby initiating a muscle contraction. Muscle contraction is initiated with the depolarization of the sarcolemma caused by the sodium ions entrance through the sodium channels associated with the acetylcholine receptors. Propagation of an action potential along the sarcolemma enters the T-tubules. The T-tubules carry the action potential into the interior of the cell, which triggers the opening of calcium channels in the membrane of the adjacent sarcoplasmic reticulum, causing calcium to diffuse out of the sarcoplasmic reticulum and into the sarcoplasm. This action initiates the contraction of the muscle fiber. Once the muscle fiber is stimulated by the motor neuron, actin and myosin, protein filaments within the skeletal muscle fiber slide past each other to produce a contraction. The sliding filament theory is the most widely accepted explanation for how this occurs. Now I will explain the cycle of sliding filament theory. On this stage, ATP is hydrolyzed and coax the myosin molecule at the hinge. The next stage, actin and myosin bind. Then the next stage, phosphate inorganic dissociates from the myosin and myosin moves to its relaxed position. And this results in a power stroke, and actin is pulled toward the center of the sarcomere. ADP dissociates from myosin at the end of the power stroke. Here. Then ATP binds to the myosin head and breaks the actin-myosin attachment. We see this on this picture. And if ATP is available and the calcium 2 level in the sarcoplasm is high, then the contraction cycle will continue over and over.